Just hold on. Excuse me. So we're working on the system.
Oh, okay.
So,
Yeah.
don't worry
I'm
about it.
sorry.
Thank you. So you're all set up? Computer? Yeah. Okay. Well, let me grab the
I mean, it was connected
Yeah.
to start. Uh-oh. Okay, get the guy.
Okay.
I have no idea.
Once a year, I do a talk with my dad for a prostate cancer survivor. So they're just yeah. Especially if they have the disease, I'm definitely really interested. Thank you for giving me this chance, Pat.
Just give it a second to warm up.
It's dead at the moment. It's muted.
No, the feedback's not working.
Did you reboot the switch?
it's
There it is.
working.
It is on.
Yeah.
Yeah. It's weird because 2 hours ago we did Winters checking it.
The mute states are wrong now.
We're ready. If you'd like to put your mic up.
Or
I can
You don't want
yeah,
to
yeah,
laugh.
go for it.
So I apologize, we're gonna have to mic you with two. Yes. You are special. So Passegué.
Sometimes you have to. And here is the second one. Perfect.
Our speaker today is Panasonic. Okay, so this is ready to go. Oh, that looks better. Good afternoon. Thank you so much for joining us today for the 2016 Roy Walford lectureship. Some of you might actually know Roy Walford. He was a distinguished member of our department in the university community for decades. One of the truly creative people that I've known in my lifetime in academics. He did very probing work on the relationship of metabolism to aging. He did important work on the uncovering of HLA genetics and its application to disease. And in many other ways, he contributed to the creative community of Los Angeles, which it's a wonderful thing for you to look at if you want to Google some of his activities. So we're very fortunate that the family had endowed a lectureship in his memory to capture that spirit of creativity and the theme of aging. This year we're very privileged to capture that spirit with our lecturer for the year, Emmanuelle Passegué. She is a distinguished member of the UCSF community and is a professor in medicine and in their Broad Institute for Regenerative Medicine and Stem Cell Research. She started her career in training at the University of Paris 11, and then did postdoctoral work with Edwin Wagner and with Irv Weissman. She then joined the faculty at UCSF and had an absolutely meteoric career, contributing to some of the really foundational work that we now have on the programming of hematopoietic stem cells and how that elaborates in our understanding about leukemogenesis and to aging. So we're very privileged to have her today to teach us, and thank you very much for coming to join us in Los Angeles.
Thanks a lot, Dr. Brown. Thanks a lot. It's really my pleasure and an honor to be here for this lecture. And considering the topic of the Endowed lectureship, I have decided to focus what I will present to the work done on aging hematopoietic stem cells that we tend to represent as this poor old hematopoietic stem cell on a walker, and tell you what we have learned over the last 5-6 years in the lab about what goes wrong into this aging hematopoietic stem cell and how it connects to the blood system. But before, since I didn't know about the background of the attendee, I will give a few words, very general introduction on aging. So aging, I think everybody past 40 years of age is starting to think about, because it's become much more concrete than before. And when you look at this very graceful elderly lady, you can realize that it's a very complex and gradual process where almost all aspects of organismal physiology are affected. And it's really, put in simple terms of tissue maintenance and repair, it's an imbalance between the number of accumulated damage and the capacity of repair that the tissue has to fix the damage and to let the tissue work properly. So, and this big, didn't bring us to immediately to stem cell, which are really the engine of tissue repair and maintenance in the entire body, and which can really be the unifying feature between this whole slew of disease of age-related deregulation, which affects the brain with decreased cognitive function, the blood system with immune deficiency, disease and increased rate disease of the blood vessels that was supposed to be, bone fragility, muscle atrophy, skin wrinkling, cardiac dysfunction. And among this spectrum, the most killing of all this age-related deterioration is really cardiac dysfunction. So stem cells per se are really now being put in the map of this hallmark of aging in this kind of like landmark paper, which got published in 2013 on the footstep of the hallmark of cancer development, where they really tried to summarize in this kind of like organized diagram the common mechanism, dysregulated mechanism, which can cause deterioration in the cell, fit into altered cellular function, and as a consequence lead to stem cell deregulation. And stem cell attrition or dysfunction is really one of the key unifying features of aging process at the organismal level. So when we focus on aging research, what are we looking at exactly? So if you are in the techie Bay Area and with Google and their Calico initiative, Mark Zuckerberg has already announced that the person which will live 200 years are already born today. And that's really a tall order about longevity, making the human species live longer. Actually, that has not been shown to be the case. There is an increase in life expectancy at the level of the human population. It's mostly driven by two facts: a large decrease in infant mortality in developing countries, and improving health care, and therefore the duration of the survival towards the later age. And that's what has extended human lifespan at the level of the population, but the limit of the species in terms of longevity has not changed. We still live at most around 110 years, which is close to the older recorded human being. So what really aging research in our day and age is about is really a matter of health span. We live this 180, 110 years at most for the centenarian and above. But the problem is that the onset of this age-related disease I have indicated is among us towards this later age. And past 50, 60, you really see all the curve of age-related disease increasing exponentially. So aging is the key driving factor of a lot of these late stage disease. So the goal is to really maintain an healthier population longer. And I was just mentioning to someone that in pure terms of economy, and if you take the call art fact-driven, number-driven. One way to solve the big problem, the big deficit of the healthcare system, is to basically make the people living 6 months less. So the cost of maintaining life for the last 6 months is extraordinarily high. So really decreasing disease onset, maintaining longer health span, it's a very important healthcare goal at the moment. So that brings me to my favorite tissue, the blood system. What do we know about blood aging, and what can we do about blood aging? So, but first, before going into the detail of hematopoietic stem cell and all the different blood cell population, which are clearly circulating through the blood, I want to say that the blood goes beyond that just the hematopoietic system. It's really a vector of information within the entire body, which distributes through every organ, cytokine, inflammatory molecule, metabolite, amino acid, vitamin, minerals, etc. And in the context of aging research, there is a lot of excitement at the moment in the community about rejuvenating factors which are present in the blood plasma and which have been shown to be effective at altering some feature of aging and are restoring some regenerative property in certain stem cell population. The one which have per se very limited regenerative potential, like neurostem cell and cognition, or muscle repair upon acute injury. There is as well pro-aging factor which need to be downplayed to limit general inflammation and other aspects of organismal aging. So the blood is a very important organ for physiological health, and my focus in the lab are really on the blood cell. So here is one of the classical hematopoietic tree with a blood-forming hematopoietic stem cell really sitting at the tip of this hierarchy. It's a mother of all blood cells, which are produced, all the mature blood cells, through this very complex series of intermediate population, progenitor population specialized towards the myeloid lineage. And here in the movie, what you saw was the output from one hematopoietic stem cell put on a stroma layer of OP9 with myeloid differentiation cytokine and let go towards the myeloid differentiation. It illustrates the potentiality, the regenerative power of one hematopoietic stem cell to give rise to this vast amount of myeloid cell. I am not going to talk about too much about the lymphoid arm of the pathway and differentiation toward B cell, T cell, the whole adaptive immune system, which is much more difficult to model as simply in a dish. So we live a fascinating time in the blood field because there is a lot of new techniques which are coming to play to understand the function of the hematopoietic stem cell, and there is raging battle about the role, the marker, the purity, what are stem cells? Are stem cells really contributing to producing blood in native condition when you are not doing transplantation, and so forth. So we are gaining a lot of insight about what it is to be a stem cell and what stem cells do. And my take, which is shared by some of the community, maybe not every stem cell scientist, HSC scientist at the moment, is that when we think about hematopoietic stem cell, we think about a continuum of state and a continuum of metabolic activity, and this continuum of activity doesn't necessarily perfectly match with surface marker. So we are fortunate in the blood field to have what is called flow cytometry, surface marker staining, and we can get on lineage negative cells which do not express any of the mature blood cell, get on c-KIT-SCA1 double positive population, FLIC2 minus 150 positive, separate away with 48 different subset of multipotent progenitor. I will not bore you more with flow cytometry. Actually, is one of my few slides where I show one of these FACS plot. But within this continuum here, this HSC MPP1, HSC short-term, intermediate term, this is really what we are looking at, a continuum of activity which are driving stem cell HSC biology. The most quiescent hematopoietic stem cell is kept in a very glycolytic stage, basically maintain its mitochondria activity extremely low, and that's the engrafting stem cell. That's the one you want for bone marrow transplantation, for clinical application. But then when a cell makes fate decision, it activates. And I will go back to what this activation is on the second part of my talk, where we are working a lot on the link between autophagy and mitochondria biology. But you need to activate your stem cell, and in this activated stage, that's when you make fate decision and you become lineage prime. And then you have the mechanism which return this activated cell into the quiescent phase. So this is really the life cycle of a stem cell: quiescent, engrafting, activated, OXPHOS-driven metabolism, non-engrafting cell, and the cell goes back and forth within this continuum of surface marker. And then you make fate decision, and what we recently published, and I'm not going to go into detail, is that the bias towards certain type of blood cell, the megakaryocytic lineage in particular, is due to a production of a specific lineage bias type of multipotent progenitor cell. We are not in the stem cell compartment. These cells do not self-renew, engraft very poorly, but are very good at making MEG. These MPP3 are extremely good at making granulocyte macrophage. These MPP4 very good at giving rise to B and T cell. They can all be reprogrammed through a combination of cell extrinsic and cell intrinsic effect to give rise to the other lineage, but they are already committed to certain fate. And they are not the stem cell compartment. They are here expressing 48, FLIC2, and so forth. So what happened with age? Solely looking at the stem cell compartment, what we see in all mice, and when I'm talking about all mice, I'm talking about one genetic background, C57 black 6, and I'm talking about mice which are 24 to 30 months of age. So these are almost geriatric mice. These are more like your 70, 80-year-old individual rather than a 50, 60-year-old, which is more mimic in an 18-month-old animal. And when we talk about young mice, we are talking about 6 to 12-week-old, at most 2 months of age, young mice. And that's the comparison which is made, that the pattern, the flow cytometry pattern at the level of the stem cell compartment, where you can see this massive expansion of the old hematopoietic stem cell population that, the massive expansion of the stem cell population in an old mice, which by kind of like a language license, I will call old stem cell. But we have no idea of their division history. We just know that they are stem cell in an old animal, vastly expanded in numbers, that's quantified here in frequency, that you can see that during the first year of age, the stem cell population is constrained, maintained at a small level, but then by 18 months old, you start to see an increase, but in these geriatric mice, the expansion of the stem cell compartment is way bigger. And when you functionally test this stem cell, through transplantation, which is in the blood field, the only true functional test that we have, where we see the regenerative potential of this cell, you can see that they are hugely dysfunctional, this old stem cell. When they are asked to repopulate the blood system of a young mice, they engraft, so their total chimerism is way lower, and they are still able to make myeloid cells, which are the hatch bar, but they completely fail at making lymphoid cells. And that's really the basis of this myeloid bias old hematopoiesis, which is in fact an incapability to maintain the lymphoid lineage and a lower but still preserved capability of producing myeloid cell. So accumulation of poorly engrafting stem cell is a key point of this aging blood system in terms of the stem cell compartment, and a strong hypothesis, still an hypothesis at this point, explaining the rest of the feature of an aging blood system, which are decline in immunocompetence, inability to maintain a functioning B cell compartment, a greater propensity to anemia, increased production of myeloid cell, which lead to this very high predisposition to a whole range of myeloid disease, myeloproliferative neoplasm, myelodysplastic syndrome, and bone marrow failure disease. So a lot of drivers of stem cell aging have been proposed, and they are all over this pie chart of what has as well been identified as driver of the aging process in general. Epigenetic deregulation, metabolic dysfunction, mitochondrial problem related to the SIRT gene, loss of cell polarity, DNA damage, telomere shortening. And what I would really want to focus on is our contribution to this list of drivers with our finding on replication stress and autophagy. So, long time ago, in the lifetime of a grad student, that's almost ancient history, in 2011, we published our findings that hematopoietic stem cells, thinking about the young organisms, are extremely protected in terms of their response to a DNA damaging agent like irradiation, in the sense that their mitochondria express, there is a lot of BCL2 pro-survival factors to prevent mitochondrial-driven apoptosis. So the threshold of pro-survival gene expressed in hematopoietic stem cell is way higher than in other mature blood cells within the myeloid lineage. I'm not talking again about the lymphoid lineage. And as a consequence, you need a much higher threshold of p53-driven pro-apoptotic gene to kill an hematopoietic stem cell versus more committed progenitor cell. But the counterpart of this quiescent stem cell being very protected is that it's quiescent. So when you have a DNA damaging insult caused by irradiation, double-strand break, the only way for this surviving stem cell to repair the damage is to use error-prone non-homologous unjoining type of repair, which is per se an extremely efficient repair mechanism, but extremely unprecise. It's basically a quick and dirty way to fix back two broken pieces of DNA. It does that by shooing away some of the end. You get microdeletion. You can get insertion if there is a little bit of I'm blanking on the name, the enzymatic activity, which we feel overhang. Or you can get translocation when it joins different pieces of two different chromosomes, therefore driving mutation and loss of function in the stem cell compartment due to misrepair DNA damage. And that we can show that very clearly experimentally by taking this quiescent stem cell out of their niche, irradiating them right away, and looking at the consequence of DNA repair. It's extremely mutagenic type of DNA repair. You can prevent, in large extent, this mutagenic DNA repair by forcing the stem cell to cycle. When you stimulate their proliferation, then they are totally competent to use the high-fidelity homologous recombination to fix the DNA damage, and that significantly limit the number of mutations that we can detect. So this is a perfect system to ensure the survival of the cell using a repair mechanism which allows the cell to still make progeny, but over time can lead to a lot of this aging-associated accumulation of mutation, and what has become very clear from a recent human study, which is clonal hematopoiesis. The fact that over time, even not in the context of frank disease development, you have mutated stem cells which contribute to blood production, and that happen with very high frequency, 7% of the general population, based on recent study, has clonal hematopoiesis as stem cells which carry mutation in important epigenetic remodeler. So this type of strategy is an example of antagonistic pleiotropic effect, which is one of the key aging theory, which is the wiring of the system to produce, to maintain the health of the tissue during the reproductive age of the species. So in mice, it's in young mice. In human, is young human being. So you don't want to lose your stem cell because they produce the blood. So therefore, you use this type of strategy to maintain blood production. But the downside of it is past the reproductive age, you have the mistake made by this type of repair of strategy, which now accumulate and drive cancer, age-related disease, which are happening in the individual, which are not needed for the species since they are past their reproductive age. Fairly depressive notion, I have to say, but still a very nice, efficient mechanism to maintain blood production at the expense of genomic stability. So you can think, well, it's enough to induce cycling to prevent this type of mutation. Therefore, maybe inducing cycling in all stem cells will be a way to avoid this kind of like clonal hematopoiesis and accumulation of a background of mutation. And the answer to that is not. No, because all stem cells, when they cycle, have a lot of other problems. So that's a story we published last year, which deals with this replication stress, which is a key driver for stem cell exhaustion and the impaired engraftment, which is characteristic from this whole stem cell, and lead as well to ribosomal stress and other aspects of deregulate biology. So it's a complex story. And the easiest way I ever found to explain it is to do it with picture. It has to do with stem cell activation. When the cell goes from quiescent phase of the cell cycle, not proliferating, to S phase, where they need to replicate their DNA before dividing. So what we found is that all stem cells have way lower expression of not just MCM4, but the entire family of DNA helicase mini-chromosome maintenance. So it's a DNA helicase, which is part of the pre-replication complex, which sit in front of the replication fork and basically unwind the DNA to allow the progression of the replication fork and the duplication of the DNA during S phase. As a consequence, there is a lot of replication forks problem in all stem cell. Instead of having this nice active origin of replication, which is labeled in red, surrounded by extending replication origin in green, which are very symmetric, we see much less active origin of replication in all stem cell. They need to replicate a bigger DNA territory than in the young because there is no activation of backup origin of replication or the normal number of origin of replication. And we have a lot of asymmetric replication forks, which indicate a fork collapse or stalling at this end. So lots of problem replicating the DNA, leading to DNA damage when the cells are in S phase, where we see a lot of gamma H2AX positive DNA damage foci, which co-localize with 53 BP1, indicating it's indeed a DNA damage foci, or phospho-CHIK1, which is a marker indicating activated ATR pathway, which is the kinase involved in replication stress. If you extend this whole stem cell for several days in culture, as a consequence of this replication stress, you see the typical feature of chromosomal gaps and break associated with this problem in replicating DNA. So this is in itself extremely mutagenic, leading to mutation as well as loss of function in the affected cell. And we think that exactly what happened when you force stem cell through what is really a replication challenge, which is transplantation. The cell just accumulate too many replication errors and they die from this stress. But the story is not over. What we found as well is that in late S phase, you are replicating the DNA which covers the rDNA gene. So rDNA gene are tandem repeat of the same genetic sequence with a lot of insulator region, a lot of origin of replication. It's a very hard to replicate part of the genome. That's why it's done in late S phase, and the cell can progress through S phase and enter G2M and division, even with improperly replicate rDNA loci. And we can see in this whole cell a lot of co-localization of gamma H2AX DNA damage, actual stolen replication fork on rDNA gene labeled here with an rDNA fish probe. So, and then in vivo, never happened in vitro, because in vitro we cannot put back a stem cell, young or old, into quiescence. We can only activate them and they never go back to quiescence. We really need to transplant the cell, to have them in the bone marrow niche to go back into quiescence. But in vivo, in the mice, after having divided once, the old stem cell go back to quiescence. When it goes back to quiescence, it reforms the nucleoli. The nucleoli are really all the rDNA gene coming back together into this very highly structured part of the nucleus, where you have a shell of a chromatin and a lot of like epigenetic hierarchy isolating basically this rDNA gene plus no RNA and a lot of other factors. It's kind of like site used in the nucleus to store some protein, p53 is at the nucleoli. So a very important structure in the nucleus. And what we can see is that when the rDNA gene now are all compacted back into the nucleoli, they carry with them a lot of this gamma H2X signal. And they carry it with them not because there is still ongoing DNA damage, as we had in this part of the cycle, but they carry it within because the phosphatase, which normally is dephosphorylating gamma H2X, PP4C, is actually excluded from the nucleus in old cell. In young cell, quiescent, proliferating, PP4C is always nuclear, not in old cell. So another unexpected complexity of the cellular biology of an aging hematopoietic stem cell is a mislocalization of this phosphatase improperly dephosphorylating gamma H2AX mark on the rDNA gene, leading to transcriptional silencing of the rDNA gene. We are now dealing with a mark which is an histone phosphorylation, which is acting as an epigenetic mark. And as a consequence of that, not just decreased transcription of the rDNA gene, we see very clearly in all cells which have this gamma H2AX mark, decreased protein translation. So that's the part which is labeled non-canonical function of gamma H2AX and ribosomal stress in the model that I just showed you. So we really went full circle with the biology of this aging stem cell, uncovering two very deregulated features, loss of MCM expression. We do not know why level of MCM are decreased with age in this stem cell. Is that a direct transcriptional deregulation? Is that an epigenetic consequence? A lot more work. This is a call for postdoc, if someone is interested by this question, to come to my lab. And we don't know why the phosphatase is mislocalized. So we found it for one phosphatase on one particular target. But if you think more broadly about cell biology, you can imagine that this change in phosphatase localization activity may affect a lot more other signaling pathway, because phosphatase are really used to terminate some signaling activity. So you might imagine a lot of more associated deregulation. So altogether, one of the question coming out of this work is really, will decreasing replication stress in all stem cell rejuvenate the blood system? If we can prevent that, maintain proper replication fork, avoid the DNA damage part and the associated ribosomal stress part, will be maintained healthier whole blood stem cell, which will be able to regenerate better and maintain an healthier blood production. So that's really the upcoming outstanding question coming out of this work. We as well use the now well-famous parabiosis approach has become coming back into the literature over the last 10 years as a way to identify pro-aging or rejuvenative factor. It's a very, very old technique, which is surgical connection between two mice, either young, which are isochronic, or young, old, heterochronic, or old, isochronic, old, where you basically just surgically link together the skin of these mice, and over a week time that allows them to share a common blood circulation. without sharing any other common organ. So it's really making twin mice and seeing the effect of the young blood into an old mice, which is a rejuvenating effect, or the effect of an old blood into a young mice, which is a pro-aging effect. So we use this approach to look at, is this exchange of blood, the cell, the plasma, had any effect on the hematopoietic stem cell? And unfortunately, it didn't turn out to be the case. We didn't see any change, no change in the frequency of LSK. That's just looking at the whole stem and progenitor population. Very elevated in all iso mice, still elevated in the old parabian partner of this heterochronic pair, compared to the young mice, which have low stem cell number, and that's not affected by the whole blood. Upon transplantation, we have the exact same pattern, meaning all stem cell engraft poorly, the old stem cell from the old parabian is not changed, the young stem cell from the young parabian is similar to the young iso. So we have absolutely no pro-aging rejuvenation effect on the blood stem cell. And our replication stress feature, which has this delay in division kinetics that you can see at the single cell level by monitoring the percent of cell which complete first division, and there is roughly a 4-hour delay in this whole cell, is absolutely not rescue in this heterochronic parabasis mice, as well as what I just present to you, this gamma H2AX fibrillary co-staining indicating as a residual effect of replication stress at the nucleoli is absolutely not changed in this whole heterochronic mice. So these are definitely, this replication stress feature are definitely cell-intrinsic feature of an old hematopoietic stem cell, and they are not affected by change in circulating factor coming from parabiosis with a young animal. We are now confirming this result directly with plasma injection, which is really the needed confirmatory experiment. So what we found as well a couple of years ago is that with age, another survival mechanism, which is very active in a young stem cell, is again hijacked in this vastly expanded population of all cells. And that has to do with mechanisms to survive metabolic stress. What we show is that young stem cells are extremely poised to respond to metabolic starvation. And by metabolic starvation in the blood field, I'm not talking about glucose removal or amino acid removal. I am simply talking about cytokine deprivation, which is a way we induce starvation, metabolic stress, in this young cell. So cytokine deprivation trigger right away an autophagic response, which is double membrane engulfing cellular component within the cell and degrading organelles macromolecule to release building block and energy to allow the cell to survive this metabolic stress period. It's a short-term survival strategy. You cannot eat yourself up and hope to survive, so there is always a trade-off. You can use autophagy as an acute stress response mechanism, which allow to avoid survival in starving condition, and that's exactly what the young stem cells are geared to do because of their very enhanced and very active FOXO3A transcription factor, maintaining this pro-autophagic gene expression profile in young stem cell. So what we see with age is that this inducible autophagic response in response to stress is still active, is still there. But in this vastly expanded population of all stem cells, we now see very clearly figure of autophagy, basically an activated basal autophagic level that we think is really driven by this aging bone marrow niche, this changing our microenvironment. And we demonstrate that this basal autophagy was very essential for the survival of all stem cells to mitigate an energy crisis. If we block autophagy, a lot of the old stem cells die. If we provide them with a permeable form of pyruvate, we rescue the death caused by the blockade of autophagy. So we conclude from this study that this activated basal autophagy that we observe with age was a mechanism to contribute to the expansion of this pool of dysfunctional old stem cells. So this was 3 years ago now, and I can say that it's mostly wrong by now. So because when we did this work, we really got dazzled by this picture, where these are an electron microscopy of an old hematopoietic stem cell, where you see this very clear autophagic vesicle in the cytoplasm of the cell, which you never see at all in a young, except when you cytokine deprive the young stem cell. So we focus on this autophagy activated cell. But then the terrific PhD student in my lab went back and looked at the ratio of cell having this activated autophagic response and cell without autophagic response. And actually, over 3 different preparations of cell for electron microscopy analysis, he found very matching result, which is roughly a third of the cell have an activated autophagic response, while 2/3 of the cell do not. So in parallel, he aged a large cohort of autophagy reporter mice, which are this GFP-LC3 reporter line, where there is a conjugation between LC3, which is a very important molecule for the formation of the double membrane autophagosome. So tagged with a GFP, so we can measure the fluorescence intensity on this kind of graph. So forming membrane or GFP-LC3 not used for an autophagic response will fluoresce very bright. And that's what we see in young stem cell, very high level of GFP. When you are actually an autophagic flux, you have GFP-LC3 on both sides of the autophagosome membrane. And actually, the GFP-LC3 molecule within the autophagosome, when it fused with the lysosome, got destroyed, quenched for its fluorescence. And you have a shift downward of GFP-LC3 intensity, which indicate an active autophagy. When you overlay the GFP-LC3 level in young and old stem cell, you can see that, again, taking the young as a cutoff, you can see that roughly 30% of the old stem cell population have lower GFP-LC3 level, indicating ongoing autophagic flux compared to the compared to the 70% of the rest of population, which has this autophagy low, similar level to the young stem cell. So this reporter now give us the tools to be able to isolate subset of autophagy activated and autophagy suppressed old stem cell, and to functionally test them for their ability to regenerate the blood system using this transplantation approach that I have already mentioned. So we isolate 250 old stem cells from each of these subsets using this 33% cutoff and transplant them in young recipient mice. And what we can see is that the autophagy incompetent subset, the autophagy low, which is actually the GFP high, is extremely poor at engrafting. It does produce cell at 1 month, and then the whole system crash. Versus the autophagy high is way better at providing blood regeneration. It's a little bit less myeloid biased than the autophagy low, and it's certainly much better at regenerating the stem cell compartment in this transplanted animal at 4 months post-transplantation, where the autophagy low stem cell give rise to almost no progeny, while the autophagy high give rise to a very robust stem cell compartment. And when we look back at the level of GFP-LC3 in this regenerate stem cell, we can see that the difference are somewhat persisting. So the blood feature become exacerbated upon secondary transplantation. I am not showing you the data. You isolate this like few remaining stem cell here, same number of these better engrafting stem cell. We transplant, this difference become exacerbated, but the difference in actual GFPLC3 level got attenuated, meaning you select for still the fetus, even in this context, of the whole stem cell. But the result of this experiment are really demonstrating that the autophagy I cells, the one which have this large autophagic vesicle, are the most healthier of the whole cell being present. And that go beyond just that functionality in transplantation, because when we look at our feature of replication stress, we can see that the autophagy incompetent, this GFPLC3I cell, have the feature of replication stress, the gamma H2X at the nucleoli, versus the autophagy I, the healthier whole stem cell, do not. And you can always argue that this is just stochastic, because it's a Gaussian distribution. You have always a low and an high. So we did the same experiment with young stem cell using the same type of 30% cutoff. And in the case of the young mice, there is absolutely no difference in functionality of the high and the low GFP, indicating that here, this kind of like separation based on GFP level doesn't reflect actual different functional subset of cell, which we see emerging in the old context. So how we reconcile that? So the next step of the analysis was really to look at what autophagy deficiency was doing on stem cell biology. So to look at that, we use a conditional knockout approach, focusing on the first ubiquitination cascade, ATG involving the autophagy essential gene ATG12, 5, and 16, which is really the one, ubiquitination cascade which like lipidate LC3 and really induce the formation of the autophagosome. So you remove any of this component, you delete any of this gene, you completely block this ability to undergo macroautophagy to form this double membrane vesicle. So we use a classical approach in the blood field, which is to use an inducible MX1 Cre, cross with our flox allele of ATG12. We have the exact same result with ATG5, and then induce poly-IC in an adult mice to induce deletion of autophagy, the autophagic process in adult stem cell, and perform analysis up to 8 months post-poly-IC. I will not bore you with the phenotype of these mice. What we observe is that we have preserved stem cell number in this autophagy deficient mice. We have amplification of the myeloid bias multipotent progenitor pool. We have amplification of the myeloid progenitor pool, and we actually have constitutive myeloid expansion. We have zero defect in any other lineage, which really contrasts with what has been described for acute inactivation of autophagy in erythroid cell, in T cell, in B cell. So when we do it in stem cell, we do not see problem in other lineage beside the myeloid expansion. We have maintenance of the stem cell population, which again contrasts with fetal inactivation of autophagy, mostly done with ATG7 inactivation, where the whole system crashed, so you cannot build a proper adult blood system when you block autophagy during fetal development. But the stem cell, when we take them and we transplant them, are extremely poor, very similar to old hematopoietic stem cells. They engraft poorly, and they mostly regenerate myeloid cell with a defective production of B cell. But you need stress. You need to transplant the stem cell to visualize that. So altogether indicating feature of aging in this autophagy deficient mice, which go beyond just the feature when we started doing electron microscopy analysis on this autophagy deficient stem cell. So what we saw was similar to the old HSC, which have no ability to mount an autophagic response. We see an expansion of the number of mitochondria, and especially the elongated, more active mitochondria, which are quantified here. And this is not the case in the old stem cell which have activated autophagy, or in control young stem cell which have no induction of autophagy when you don't have cytokine deprivation. We see mostly mitochondria increase. We see as well dysregulation of other organelles. We see more abundance, more vesicule, which is shown here, expanded ear, rough and ear and Golgi compartment. So beyond just mitochondria number, we are affected organelle biology, which is extremely similar between a conditional knockout young stem cell and an old stem cell incapable of mounting an autophagic response. We confirm this EM analysis with dye staining for the mitochondria, mitotacker green, which look at mitochondria mass, TMRE, mitochondria and membrane potential, DCFDA for ROS level, ER tracker, and so forth and so forth. And I'm not showing you all the control, but basically in this conditional knockout, we see all these parameters being increased as predicted from the electron microscopy analysis. When we look in the old stem cell without fractionation, just the entire population, we actually see no change. So indicating that when you look at the entire population, the feature of subset are completely disappearing. What didn't totally disappear is when we now did Sears analysis to look at metabolic flux. And these are measures of OCR, which is basically oxidative phosphorylation, OXPHOS level. So what you can probe for basal respiration, ATP production, maximum respiration by injecting different uncoupler and so forth. And I will not go to too much detail, but while the basal parameters are relatively similar between young and old stem cell, the maximum respiration capability is clearly increased in this old stem cell. We were able to do this experiment with purified old cell, and my student and a couple of postdocs killed close to 70, almost 80 mice just to get matching number of young stem cell because you really need a lot of cell. We were not able to use as pure stem cell population for the conditional knockout, so we did that on HSPC, which is a mixture of stem cell and multipotent progenitor. But again, the result is very similar basal parameter and huge increase in maximum respiration, indicating an active oxidative phosphorylation metabolism in both the old and the conditional knockout stem cell. Glycolysis, and looking now at ECA acidification rate, is not very significantly affected. Maybe slightly decreased, but none of these points are significant in the old, certainly unchanged when we look in the conditional knockout. So we are activating an oxidative metabolism without significant change on the glycolytic rate in this cell. And this really goes at the heart of what is hematopoietic stem cell activation. And you can really probe that in vitro by isolating stem cell and growing them in condition plus minus cytokine for up to 24, 25 hours to look at their metabolic parameter before the first cell division. And when you do that, you see very quickly that in cytokine condition, which are in black, you activate the TOR pathway. Phosphox 6 goes way up. You have activation of mTOR, which doesn't happen when you have no cytokine. You see, in contrast, when you have no cytokine, very quickly activation of AMPK. So mTOR prevent autophagy. When we look with GFPLC3 stem cell at the autophagic flux, we can see that with cytokine, we basically increase the GFPLC3 level, which indicate active suppression of autophagy, while when we remove cytokine, we don't activate mTOR, we activate AMKP, and we actually very quickly induce autophagy. And when we look at the type of autophagy which is happening, we can clearly see mitophagy figure, where you have all mitochondria in this autophagosome. So this leads us to the idea that stem cell activation corresponds to mitochondria activation and active suppression of autophagy. And you can see that with many different parameters, over time, cell size increase, mitochondrial mass increase, mitochondrial membrane potential increase, ROS level increase, and these ROS levels are not cytotoxic. We look a lot, there is no DNA damage associated with that, there is no induction of apoptosis. These are really signaling level of ROS, similar to what Hupal here has already shown in the fly system with induction and poising toward myeloid differentiation. So when we do the metabolic flux now on this activated stem cell, 22 hours after being exposed to cytokine, you can see this very clear ramp up of oxidative phosphorylation. So this is a normal activation part of the process, and we place really autophagy in this return to quiescence, this quick clearance of this activated mitochondria to basically revert the metabolism of this cell to a quiescent, mostly glycolytic, not oxidative phosphorylation-driven metabolism. And when you block this ability to activate autophagy, which is what we have in our conditional knockout stem cell or in a large fraction of the whole cell, you basically maintain your stem cell in this highly activated metabolic state where you can see increase in size, increase in ATP level being produced, increase in NADH level, all the metabolite and byproduct of mitochondrial activity. And in the whole context, this metabolic activation, overactivation, is really specific of the AT low subset. So we are still building with this genetic reporter mice in the separating this subset, but we can see that the increase in cell size is only in the AT low, the increase in NADH level, the ROS higher, the membrane potential, the ROS, it's really the cell which do not activate autophagy, which have this overactive metabolism. And this overactive metabolism really drive functional impairment. We see both in the conditional knockout and the whole setting the same loss of quiescent. It's not the entire population, the quiescent population, which is gone. We see basically a reduction by roughly 20% of the number of quiescent whole stem cells. So there is a progressive erosion of this quiescent subset, most likely because mitochondrial decay will allow some of the cell to return to quiescent. We see as well a very clear promyeloid differentiation feature, which we can look at by methylcellulose colony assay, where the cell, the conditional knockout cell, like young stem cell treated with BAFE or like old stem cell untreated, have basically reduced plating efficiency and reduced number of immature colony, which are reflective of a more uncommitted fate, which are the mixed and the GM type of colony. I'm not getting to a lot of detail about this assay, but you can see preservation of mature type of colony, one fate, loss of immature colony, and in the old treated with BAFE, so when we completely block autophagy, this ability to form immature colony completely disappear. So it's not a transcriptional effect. We have spent a lot of time trying to extract meaning out of microarray data, comparing conditional knockout versus control HSC. There is absolutely no statistical change in gene expression except for one, and that's ATG12, which is the gene which we inactivate. So there is literally no transcriptional change, but there is a very clear epigenetic signature. In the conditional knockout stem cell, we see now very clearly an enrichment in hypomethylated DNA region. And this hypomethylated DNA region really overlap with phosphoprotein and intracellular signaling cascade. So by removing autophagy, we are doing metabolic reprogramming, and this metabolic reprogramming will feed into an epigenetic reprogramming, which poisons the cell and which is way before any transcriptional change. And this kind of change in the cell activity is really driven by the bone marrow niche. I'm not going into a lot of detail here, but we can see that, like, going back to our parabiosis approach, we have a lot of spillover of old stem cell into the young bone marrow, into this heterochronic parabiosis mice. And when we test the fitness of old cell which stay in the old bone marrow, this is way worse than old cell which are now in the young bone marrow. So the aging bone marrow niche is really what driving this autophagic response. And that lead me to my few final model slide and the end of my talk, where we basically are learning a lot about the role of metabolism and autophagy in maintaining stem cell function. And it's really in this part of the process, this return to quiescence. That's where macroautophagy is important. And when you don't have autophagy, you don't have physiological mitochondria clearance. And I'm talking about physiological clearance because we study as well the parking2 deficient mice, which are defective in clearance of damaged mitochondria. And that's absolutely not the phenotype that we see in our ATG12 mice. Actually, parking2 deficient stem cells in our hand are totally fine stem cells. So, and this lack of mitochondrial clearance lead to an altered metabolism homeostasis, epigenetic regulation, and really trap the stem cell population in this poised stage where you have much more impetus to go down towards the granulocytic macrophage myeloid differentiation pathway and the altered loss of quiescence and per-engraftment pro-myeloid differentiation. It's as well tell us that, well, for most of the function at steady state in native condition, a stem cell without autophagy does relatively well. Yes, it's maintained, it has increased blood production, some myeloid expansion, but you still make blood, and the mice live, and they live long on this imperfect hematopoiesis. So there is clearly alternative pathway of protein recycling which are being activated. We are starting to look at microautophagy and endosomal recycling to explain that. But this is a relatively fine part of the equation. The problem starts when we induce blood regeneration. So they are not able to mount a proper regenerative response. That's where when we see exhaustion, decreased blood production and cytopenia. And that was question about the side effect of autophagy inhibition, because there is a lot of clinical trial happening at the moment, which are trying to block autophagy in tumor, and that might have a lot of unanticipated side effect on the blood system. It still remains to be determined if transient blockade of autophagy with a drug will be equivalent to genetic ablation of autophagy as we model it in our mouse model, but I will be very cautious with this kind of clinical trial. And finally, it tells us that our conclusion on aging were wrong. So effectively, the aging feature of the blood system are driven by the large fraction, 2/3 of the population, which failed to activate autophagy in this aging bone marrow niche. And these are the unhealthy old stem cells, which are myeloid biased, have the loss of regeneration potential, replication stress feature, and so forth. While the healthy old stem cells are the ones which activate autophagy and do not have this feature. So the key question now is why this aging bone marrow niche is driving this functional difference, this subset activating and other subset not activating autophagy, and can we target it for rejuvenation intervention? And towards this end, we are using a spectrum of approach. We are starting to profile this sorted autophagy high, autophagy deficient subset for RNA-seq, the exome sequencing, and so forth. We are using the panoply of life-extending intervention, which have been shown to control mTOR and to impact on autophagy, rapamycin treatment, caloric restriction, to see if we can manipulate autophagy level in all stem cell and change stem cell HSC fitness and blood parameter. And we are performing a lot of niche study to understand what aging adverse environment mean. And all of that is really for the goal of improving health span and blood production in the elderly, which really drives the point of trying to find intervention which can be given preemptively to people, to elderly people, in order to avoid this, the development, the accumulation of mutation. And with that, I want to stop by thanking the people in my lab which did this work. Everything I've shown you about the new data has been done by Theodore Oh, which is a terrific grad student, started together with Matt Ware, former postdoc in the lab. Joanna did all the replication stress work we published, and Siske and Genia are as well very involved in the parabiosis experiment and other aspects of our aging study. Jay Debnat is our resident expert on autophagy at UCSF. Sol Villeda is helping us with the parabiosis approach, and Ken Figueroa really did a terrific job with methylation analysis. And with that, I thank you for your attention and we'll take questions. If there is question. Yes.
So there really is a stochastic loss, a stochastic accumulation of cells which have lost the autophagy.
So I say loss of autophagy, but it's not a proper expression in the sense that we take this subset out of an old mice, we put them in vitro, and we trigger autophagy with cytokine deprivation. Both subset can induce autophagy and mount autophagic flux to the same extent. So they are not they have not lost the ability. They are just incompetent at doing autophagy, at mounting an autophagic response in vivo in the bone marrow niche, which is very good for thinking about along the line of intervention to manipulate this ratio. But we don't know yet why some activate, some do not. And so we are still waiting for our RNA-seq data. Yeah?
So in anorexia nervosa has been demonstrated to reduce liver failure through autophagy. And so could that possibly be a link between calorie restriction and longevity?
So this is a loaded question, as all these kind of questions are when you talk about an intervention, organismal aging, and restoration of some function. So, and that's what our learning process has been, taking this established intervention and trying to now focus on the blood system and hematopoietic stem cell health. None of the intervention we have looked at has changed anything for the blood system. The only change are making them worse. Example, caloric restriction. The mice look way healthier. I mean, no comparison between caloric restricted old mice and ad libitum fed old mice. So you say, good, it's a good thing for aging. We look at their blood stem cell, no difference. They are still as old as dysfunctional. We did exercise. You will argue it's kind of like the weekend warrior which go on an intense exercise. So we put all mice on the training wheel. They love to run. They run a lot. And they look better. They are less fat. You look at their cognition, their brain activity. They are smarter by all the behavioral parameters. we look at their blood system, at their hematopoietic stem cell, terrible. That's the worst situation. They are becoming activated, big wave of inflammation, their stem cell functionality plunger. So I think that's why I say these are loaded questions, because there is intervention, nutrition, which are making the organism better, but doesn't mean the effect on tissue regeneration, maintenance, and stem cell function. And I think each stem cell will be kind of different in its mechanism to respond. So I will be careful. But this being said, it's clear that exercise and caloric restriction are the best preventive approach we have so far. So please eat well, exercise. I'm trying. It's relatively unsuccessful so far. Yeah.
Those lucky mice in the intervention study are never exposed to any pathogens, and I would imagine if they tried that, the effect of the bone marrow issue would become evident, and they probably wouldn't live longer. That's my guess.
Yes, yes. But we try to keep our animal in clean condition, and for this long-term study, it's even more the case. And that's now the point, which is we started this study with aging ourselves our mice, and we got way more severe features than what we get with the NIA aging colony, because the NIA is aging mice on a lighter, less caloric-rich diet, and the old mice we are getting from the NIA are slim, good-looking old mice. And I mean, they are really your healthy aging population, not your potato couch population. And so you really are doing aging study on selected subset of mice. So I agree, there is a lot more to be integrated into this kind of aging research, infection, the challenge, the stem time that you give intervention. All of that require an infrastructure and funding, so which we don't have at the moment so much. Mm-hmm.
I just have a question for the length of time of your parabiosis.
Yes. So we did what has been reported, which is 1 month. So, and we did 2 types of age match. So 3 months young, 18 months old, 3 months young, 24 months old. The problem is the older you get the old mice, the less survival, so it really goes down. So we lost almost all of our heterochronic old when they were 24 months, and a large fraction of our isochronic. And in none of this situation we saw rescue. So we are still testing the cross-engraftment of the old cell in the young bone marrow, because there is a lot of, as I say, spillover. So there is absolutely no young stem cell entering the old bone marrow niche, like bad environment. But there is old cell in the young bone marrow. And we have not seen any rejuvenation on this old stem cell going to the young bone marrow niche. And that might be compounded by the fact that they are early immigrant, and it takes stem cell when they home at least a month to revert back to a quiescent stage, which is engraftable. So we are trying now to make separation experiment, where we separate the young and the old, let the young now for 3-4 months, and see if the old stem cells, which are home in the young bone marrow, better. But it's a UC system, and we have not been able to get the separation protocol approved by a Cook committee. So that's where we are struggling. Mm-hmm.
2 questions with regard to the parabiosis experiments. Can you comment on GF11?
No, I don't, and I don't want. That's another loaded question.
With the experiments in the 50s, there were experiments showing that parabiosis protects animals from acute radiation syndrome.
Yeah.
So the question here is, if you challenge these animals and force basically the niche to be emptied out.
Yeah. So we did a little bit of GCSF treatment to try to see if we could force cross-engraftment by giving a mobilizing cytokine. We didn't really change anything. So we still got like roughly like 40% chimerism all cell into the young niche. Very interestingly, and that's not the way I show you the data because I show you just the frequency, but it's a displacement of the young stem cell because we are keeping the same size hematopoietic stem cell niche, meaning the old cell are killing always the young and are taking residence in the young bone marrow niche. And this was not changed by the GCSF treatment. What was changed is the survival of our mice. I mean, as soon as you start challenging, they just die as bankbuster. I mean, these are very dramatic intervention. Like there is a persisting inflammatory response associated with the parabiosis, and any other challenge, they might just die.
And then the second question is when you look at your AT high and low cells, are the AT high cells the cells that have a healthier genome? Did you look at that?
We have collected, we will get the RNA-seq data way before the exome sequencing, but we have collected material. All the problem is logistic, is you need to wait until you have a 24-month-old GFPLC3 reporter. We start with a lot of mice we age, but all these genetically modified mice age poorly compared to wild-type strain. So the age-related attrition in this cohort is humongous. So we just don't have enough mice to go quick. So it will come, but we don't have an answer. We only have the IF image for the replication stress and the Terasaki experiment done with the high and low, which shows that like the low have the feature of replication stress. So they might be loaded with mutation, or not. Or it will be the autophagy high, because when you think about the clonal hematopoiesis, you are emerging clones of stem cell which carry epigenetic remodeling enzyme. And this can be this autophagy high subset, because they are healthier, meaning they engraft better, but they might just be more competitive and already pre-mutated. So it can go many different ways. At this point, we don't know. Yeah.
So when you transplant the AT high versus low into the recipient, you look at peripheral blood and wondering, do the ratio of the lymphoid cells versus myeloid cells, they are the same?
If you are really like with wishful thinking, you can say that there is a little bit more normalization of the myeloid lymphoid ratio with autophagy high. And it's much more obvious in secondary transplantation mice than in primary. But it's not day and night. And although the autophagy high engraft better than the autophagy low, which really do very poorly, it's still not normalized to a young level. It's still in the range of what all cells are doing. So yes, but the connection to the lineage bias is not as clear as the connection to survival, self-renewal activity, and metabolic fitness, basically.
And the other question, so the ratio of the high versus low are the same in young versus old, or change?
So the way we did it was to take 33-33. Based on the young curve and the old curve. Because when we look at the fraction of cells which activate autophagy in the old, it was roughly 30% of the population when we compare to the young. But in the young, we just took 33-33. So it's different levels than what we took in the old.
I think for more questions, we should continue them afterwards. I want to thank you so much for such an interesting, provocative presentation, and thank you all for sharing this time together. Congratulations.
Thank you. My pleasure.